In the depths of the rainforests, an invisible pathogen emerged that would change our understanding of viral hemorrhagic fevers forever. Known as Ebola, it is a name that often evokes fear, but behind the headlines lies a complex biological puzzle. Today on MedSimpleTV, we're stripping away the myths and diving deep into the science of the Ebola virus. We will explore how it operates, how we fight it, and what you need to know about this global health challenge. Ebola Virus Disease, or EVD, is caused by a group of viruses within the genus Ebola virus. While there are 6 known species, 4 are known to cause disease in humans. The most famous, and deadliest, is the Zaire Ebola virus. It belongs to the Filoviridae family, named for their thread-like appearance under a microscope. Unlike common respiratory viruses, Ebola targets specific systems in the body, leading to systemic failure if not treated quickly. To understand Ebola, we have to look at the cellular level. The virus enters the body through mucous membranes or broken skin. Once inside, it uses its surface glycoproteins to attach to host cells, particularly macrophages and dendritic cells, the very sentinels of our immune system. It tricks the cell into swallowing it through a process called macropinocytosis. Once inside, it releases its genetic material, turning the cell into a factory to churn out thousands of new viral copies. One of the most dangerous aspects of Ebola is how it disables the immune response. It blocks the production of interferon, the body's natural antiviral signal. As the virus spreads unchecked, the immune system eventually overreacts in a desperate cytokine storm. This massive release of inflammatory chemicals causes blood vessels to leak, leading to the low blood pressure and organ failure that characterize the late stages of the disease. Ebola is a zoonotic virus, meaning it lives in animals before jumping to humans. Scientists believe fruit bats are the natural reservoir. Spillover occurs when humans come into contact with the blood, secretions, or organs of infected animals, often through the handling of bushmeat or through contact with infected primates. Once a single human is infected, the virus spreads through direct contact with infected bodily fluids, including blood, saliva, and sweat. After exposure, there is an incubation period where the person is not yet contagious. This typically lasts 2 to 21 days. The early symptoms are often mistaken for malaria or the flu, a sudden high fever, profound weakness, and muscle pain. This is a critical window. Because these symptoms are nonspecific, early detection requires a high level of clinical suspicion, especially in areas with known outbreaks. As the disease progresses, the symptoms become more severe. Patients develop vomiting, diarrhea, and a characteristic rash. Internally, the liver and kidneys begin to fail. While Ebola is often called a hemorrhagic fever, only about half of patients experience visible bleeding. However, internal bleeding and the loss of vascular integrity are what lead to hypovolemic shock, the primary cause of death in fatal cases. Diagnosing Ebola requires specialized laboratory facilities. The gold standard is the RT-PCR test, which looks for the virus's genetic material in a blood sample. Because the virus is highly infectious, samples must be handled in biosafety level 4 labs. Rapid diagnostic tests are also being developed to provide results in the field, which is essential for containing outbreaks in remote regions. For decades, treatment was limited to supportive care, fluids, and electrolytes. But recently, we've seen a revolution. Treatments like Inmazeb and Ebanga, which are monoclonal antibodies, have been proven to significantly increase survival rates if administered early. These drugs work by binding to the virus and preventing it from entering cells. Combined with aggressive fluid resuscitation, Ebola is no longer the automatic death sentence it once was. Prevention is our strongest weapon. The ERVEBO vaccine has been a game changer in controlling outbreaks through ring vaccination, vaccinating everyone who came into contact with an infected person. Additionally, community engagement is vital. Educating people about safe burial practices and hand hygiene can stop the spread in its tracks. Ebola remains a serious threat, but science is winning the battle. From understanding its cellular entry to developing effective vaccines, we have made incredible strides. Thank you for joining us on MedSimpleTV. If you found this breakdown helpful, subscribe, like this video, and share it to help spread accurate medical knowledge. Your support helps us keep medical science simple for everyone.